class. So I am to discuss to you about restriction enzymes, and I know for sure that you already have this one with Miss Teen in your laboratory, and you also had some examples of the analysis and also the restriction mapping, which is related, okay, to this topic. So I suggest, because the exam is already fast approaching, you have to read your books and you have to read or to utilize the internet if ever you do not understand anything about the topic, because I am aware that you understand that our exams are usually analysis questions, and it requires you to really understand the topic beyond the scope that is presented to you by your teachers. Okay? So, um, let me define first restriction enzymes. So, the layman's term or the layman's definition of restriction enzyme is that they are molecular scissors. Okay, going back to the first picture that I have shown you, as what you can see here, there's a scissor cutting what? Okay, cutting the double-stranded DNA. So that's the main purpose of restriction enzyme. Okay, it cuts the DNA, and take note, because we are talking about enzymes. Okay, let me just remind you that one of the outstanding characteristics of an enzyme is its specificity. Okay, they are very specific in nature. Okay, if we are talking about an enzyme and a substrate, okay, I hope you remember that in your clinical chemistry. Okay, the substrate has to fit perfectly to the enzyme in order for it to produce a product, in order for a reaction to proceed. Okay, so meaning to say, when enzymes do their function, they have this characteristic specificity, and it's also the same as when we are talking about restriction enzymes, or which we commonly refer to as restriction endonucleases. Okay, so anyway, talking about specificity, aside from the fact that your restriction enzymes cut your DNA, okay, you have to take note that it cuts the DNA molecule at specific points. Meaning to say, there is that nitrogen bases. There are nitrogen bases, I mean, in your DNA, okay, and it depends on the type of enzymes, okay, that will act on the DNA, okay, in cutting the DNA, to what region of that basis, okay, it will cut or it will cleave. Okay, so that's the function of your restriction enzyme. And I know in the laboratory you have already performed this one, okay, DNA manipulation. Okay, so that's one of the applications of the restriction enzymes. This is just a picture showing you the function of the restriction enzyme. So they are again molecular rulers. Sorry for that. Molecular scissors. I mean molecular scissors, which cuts the DNA in their specific points or specific sequence. So what happens is you have that restriction sites. Okay, restriction site, that is where your enzymes will cut depending on the sequences of the nitrogen bases. And again, for the uses, you have this DNA mapping, molecular cloning. I think I have mentioned this one in the previous lecture, right? Molecular cloning. Okay, and I know you have encountered this term already. Okay, remember, I have mentioned this in what topic? The cloning. Okay, our topic in, okay, amplification methods. Okay, because two of the examples that I have given you for amplification is number one, the most common that we use in the laboratory. What is it? That is, okay, that is PCR, or polymerase chain reaction. So that's a type of amplification reaction. Other than the PCR, again, as what I have mentioned before, okay, we have cloning technique. So that's also a type of amplification method. And of course, you have the gene sequencing and the protein expression. Okay? One of the things that you also have to consider about restriction enzyme is it helps in the recognition of the DNA of the host. For example, I have here a bacterium. Okay? And then of course, okay, we have here the chromosomes of the bacterium. That's the chromosome. Okay, so this is the chromosome of the bacterium. Another one, you have this structure, okay, and that is what we call as what? This is the chromosome. This one, it's outside of the chromosome. That is an extracellular, extra chromosomal structure rather. Okay, so extra chromosomal structure. So what is this one? Okay, we call it this, okay, we call it as plasmid. Meaning to say, your plasmid is an extra chromosomal DNA or RNA. Okay, extra chromosomal DNA. And since I said that we are talking about a bacterium, you have to take note that most of the time, okay, the plasmid of the bacteria are circular. Okay, so plasmid, they are DNA and usually they are circular. Okay, so that's one of the characteristics of the plasmid. Okay, so going back, as what I have said, restriction enzymes usually, okay, recognizes the DNA of the bacteria, right? Okay, and in your lecture, in your laboratory, okay, what is that one component, okay, or one structure that will help the DNA of the host, either a human host or in this case bacterial host, okay, in protecting from restriction enzyme degradation. So what is that structure? Again, found in the DNA of the host, such as bacterial cell, that protects it from degradation or cleaving or cleavage or cutting, okay, of the restriction enzyme. What is that structure? Okay, this one. Do you remember this one? This is the methyl group. Methyl group. Okay, anyway, another question. Is plasmid usually single-stranded or double-stranded DNA? Okay, plasmids are usually double-stranded, circular, extra chromosomal DNA. So that's why you also have the pairings of the nitrogen bases there. Okay? Anyway, as what I have said, okay, this methyl group class, okay, this methyl group, okay, I hope you still remember, okay, the structures of your amino acids. Okay, some of them, they have methyl group, right? So you can see this CH3. Okay, and what particular nitrogen bases, okay, when we are talking about the DNA and its structure, what particular nitrogen base contain methyl? Okay, what particular, again, nitrogen base contains methyl group in its structure? What is that one? Okay, we have thymine. Okay, thymine. Anyway, this methyl group class will tag, it will label, or it will recognize the, or it is, I mean, found in the plasmid of the host, such as bacterial cell. Okay, and because of that methyl group, because of that methyl group class, your DNA or the bacterial DNA will be protected from restriction enzyme. For example, if this bacteria or this bacterium was infected by this one. What is this one? Okay, this is a bacteriophage. Okay, bacteriophage is a virus infecting bacteria. So once the bacteriophage infect the bacterial cell, it will inject its genetic material. So the genetic material of the virus of the bacteriophage will be injected inside of the host cell, right? So this is also, for example, a DNA. And again, we have restriction enzymes like molecular rulers. Okay, let me just use another color. Okay, we have this molecular ruler, right? And as what I have said, that why am I saying molecular ruler? Sorry for that. Molecular scissors. Molecular scissors. Okay? So we have that molecular scissors class, which again will cut DNA, right? So take note, plasmids are DNA. Bacteriophage will have to inject its DNA. And of course, restriction enzyme is cleaving the DNA. So in order for the host DNA to be protected, okay, the methyl group will do its function. That's why the molecular scissors, the restriction enzyme, will cut, okay, the DNA of the bacteriophage if ever it infects the bacterial cell, but it will not cut, okay, the DNA of the bacterial cell or the plasmid of the bacterial cell because of the presence of what? Because of the presence of the methyl group. Okay, so that's one of the things that you might encounter when you are talking about restriction enzymes. Okay, so memorize this one. Okay, the history, just some significant milestones in restriction enzymes. So we have these people and also the time. Okay, these people were awarded with Nobel Prize because of their studies in restriction enzymes. Okay, so basically, what's really the definition of restriction enzymes aside from the fact that they are molecular scissors? Again, they are restriction endonucleases. Restriction endonucleases that recognize specific, take note of the term, specific base sequences, nitrogen base sequences, and break or restrict, or what's the other term? Cut the DNA polymer at the sugar phosphate backbone. And I'm going to show you the specific part where the restriction enzyme cuts the DNA. This one. Okay, in the 3' oxygen atom. So I hope you still recognize the structure of your nucleotide. So you have here, okay, this one, the ribose, right? You have here the nitrogen bases. Okay, you have here what this is one? Okay, the phosphate group. And of course, this one nucleotide is connected to another nucleotide by what bond? Okay, that is phosphodiester bonds. So meaning to say, that is the bond that will be cleaved, that will be cut by the restriction enzyme. And in what specific region of that phosphodiester bonds in the phosphate backbone, in the sugar phosphate backbone of the DNA. So it's the 3' oxygen atom. So you have here the enlarged version. So this is 1, 2, this is the 3' oxygen atom. So meaning to say, the restriction enzyme will have to have its function here. And the phosphate atom is broken. So that is the specific region, okay, of the action of your restriction enzyme. So it cleaves, it breaks, it restricts, okay, it cuts the DNA at the 3' oxygen atom, breaking the phosphate, okay, phosphate bonds, okay, or the phosphate region. Okay, and where do we isolate normally the restriction enzymes? Normally, we obtain them or we isolate them from bacteria, and they function as part of the primitive defense system to cleave the foreign DNA entering the bacterial cell. Just what I have explained to you. This foreign DNA is coming from what virus? Okay, again, this usually comes from a bacteriophage. So they are defense system because again, they cleave the foreign DNA. They will only cleave the DNA of the bacteriophage, but they will protect the DNA of the host cell. Because of what structure again? Because of the methyl group found in the host's DNA. Okay? So these are also examples of enzymes. I know you have already encountered this in your laboratory. So like BAMH1, so that is coming from Bacillus amyloliquefaciens. So later I will show you again the nomenclature of the restriction enzymes that for sure you have already memorized. So they have that nomenclature, of course, so that the people that in the laboratory will not have a hard time. Okay, will not have a hard time identifying the specific enzymes and of course their functions. Okay, for example, this one. So the Hay3. Okay, what nitrogen bases, okay, does it recognize? Okay, you have here an example of the sequences. Okay, for example, we have here um, this one. Okay, so GGCC. Okay, CCGG. So meaning to say, this Hey3 recognizes this sequence class. And what do you notice here? GGCC. GGCC. Again, we term it as palindromic. When you say palindromic, reading it forward or backward, you will get the same letters or words. Like for example, your name is Hannah. Hannah with an H. 2H. So when you read it backward, Hannah. Or you have this word, level. So it's still the same, right? Or if somebody calls you Madam. Okay, so Madam. Okay, so that's still, okay, palindromic. Okay, just like in April, I think it starts in April 20 last time, this one. When you read the numbers backward, they are having palindromes. Okay, so that's just an example in order for you to really understand the concept of these palindromes and why they are important in restriction enzymes also. Because normally, restriction enzymes recognize or cut the nitrogen bases showing palindromes. Okay? So once it happens, once it recognizes that palindromes, it will cut it at a specific part. It's either in between the G and the C or in between of the two G. It depends really on the type of restriction and the nucleases that you are talking about. Okay, in this case, it cuts between the G and the C, right? So if that happens, what will be produced here? Okay, blunt ends. Blunt ends. Meaning to say, if this is cut, you can insert any sequences. Okay, any sequences. Because you do not have that template, right? Because the nucleotide, the nitrogen bases, I mean, are cut into equal parts. Okay, that's why the sticky ends class, as what you have noticed here, okay, this is the one that we commonly use in the laboratory because they are not cut equally. They're not symmetrical. Okay? So sticky ends is what you have noticed here. It also cut the DNA nitrogen bases at a specific location. Like in this case, okay, for example, it is between G and A. So it's between G and A. So what is produced here is this one. G, okay, and also here. So meaning to say, because you have sticky ends here, you cannot just put any sequences, right? You cannot just place any random nitrogen bases sequences. You need to make sure that the nitrogen bases that you are using is paired to this kind of sequences also. So that's the advantage of sticky ends. That's why it's the most commonly used in the laboratory. Okay, because we can just cut specific sequences from the source DNA and then transfer it to a vector. Okay, having that same sequence. Okay, or having that same pair. So that's one of the advantages of sticky ends. Okay, so for the biological role of the restriction enzymes or restriction endonucleases class, okay, it's for the restriction modification system. Okay, and take note class, okay, the main function also of restriction enzyme is it prevents as what I have mentioned, it prevents the replication of phages, bacteriophages, by again cleaving the DNA of the phages at a specific part, okay, a specific parts, okay, or specific sites. And another thing, as what I have said, I have mentioned this one, it's the methyl group that will protect the host DNA in by or it prevents the 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 recognition of the I mean the I get lost. Okay, the methyl group class or the methylation will produce the recognition by the restriction enzyme because the recognition the restriction enzyme class will not cut, okay, the DNA again of the host cell because of the presence of methyl. Okay, so this one class again, the host DNA is protected by these enzymes, methylases, okay, because those methylases will be the one to add the methyl group, which can be found in the host DNA. And take note, the nitrogen bases to which methylation occurs, okay, to which methylation occurs, where adenine and cytosine. Why is it that thymine was not mentioned here? Okay, because thymine contains methyl group. And why is it that uracil was not included? Because uracil is only found in the RNA and we cannot find uracil in DNA. Okay? This one class, this just shows you again, okay, the methyl group protecting the host DNA or the host plasmid. So this is the bacteria class. This is the bacterium, okay, and this is the host DNA. I mean the phage DNA. So the restriction enzyme again will cleave, okay, that DNA, leaving this host DNA protected because of methylation. This is just again showing you a different picture but still having the same principle. Okay, so these are the different types of enzymes and their recognition sequence. And as what you have noticed, we have here, okay, we have here this slash. Okay, so that's just specify in what nitrogen base, okay, will the enzyme act on. Like echo R1, it's between G and A. BAM-H1 is between two Gs. Okay, HIN3 is between two adenines. Okay, PST1 it is between A and G, and PME1 it's between T and A. Okay, memorize that one. Next one, we have different types of restriction enzymes. Actually, you have four, but for the sake of this discussion, we will only differentiate the three restriction enzymes. Okay? So for type 1 restriction enzyme class, they have not only methylase activity, but they also have this nuclease activity in one single enzyme. Okay? And they bind to host specific DNA sites of 4 to 6 base pairs separated by 6 to 8 base pairs and containing methylated what nitrogen base adenine. And I know that in your laboratory, okay, the number of base pairs, okay, was mentioned. How many base pairs, okay, it will act on, or it will count first, or it will scan. Okay, it will scan first 100 base pairs before, okay, it reaches to the specific parts where it can cleave or cut the DNA. So 1000 base pairs. Okay, that is for type 1 restriction enzyme. So for example, we have the echo K or the echo K12. Okay, you might be confused. Okay, what is this N? Okay, again, these are sorry for that. Okay, this one ATGC. This one we have GC, ATGC, and GC. So this one class, these are the nucleotides to be added. Okay, that represents non-specific nucleotides, and of course usually in type 1 restriction enzymes, again, adenines are methylated. Okay, the ends here represent the sequences that you will add in the sticky ends. Okay, for type 2 restriction enzymes, take note class, I have mentioned palindromes, and you have to take note that among the types of restriction enzymes, it's usually the type 2 restriction enzymes that exhibit recognition, okay, of palindromes. Okay, and take note that also that usually the type 2 restriction enzymes are usually found in all prokaryotes, but sometimes they can also be found in eukaryotes. But technically, all prokaryotes will have this type 2 restriction enzymes. And since usually we are manipulating the bacteria in the laboratory, we are commonly using or frequently using this type 2 restriction enzymes. However, unlike your type 1, they do not have inherent methylation activity in the same molecule as the nuclease activity. Because in type 1 class, you have that nuclease activity and methylase activity in one enzyme, right? But this one, it's not like that. Okay? And again, as what I have said, they are usually recognizing palindromes, and aside from that, they are usually producing what type of ends? Blunt ends or sticky ends? Okay, blunt ends. Okay, they bind the simple dimers to their symmetrical DNA recognition sites. So symmetrical because they cut the nitrogen bases into equal parts. However, not all type 2 restriction enzymes produce blunt ends. Okay, so bilateral symmetry. Just like this one, echo R1 again. Okay, the SMA1, the PST1 also. Okay, next one, class. This is the reason why type 2 restriction enzymes are commonly used in the laboratory, because they have, again, normally and usually bilateral symmetry. So although type 2 restriction enzymes work with bilateral symmetry, their patterns of double-stranded, okay, breaks, double-stranded breaks differ, okay, and some enzymes cut the duplex with staggered separation, or that is also what we call as overhangs. So it's not all the time that type 2 will create blunt ends. You can have the staggered or overhangs or the sticky ends. Another thing, some enzymes again cut the duplex with a staggered separation at the recognition site, leaving 2 to 4 base single-stranded overhangs at the ends of the DNA. And the single-stranded ends can hybridize. This is the reason. So the single-stranded ends can hybridize with complementary, okay, complementary ends on the other DNA fragments, directing the efficient joining of cut ends. And because of their ability to form what? Hydrogen bonds with complementary overhangs, these cut are said to produce sticky ends. Because, and again, unlike your blunt ends class, okay, they are cut into symmetrical nitrogen bases, right? So they are equal. Okay, however, again, the type 2 restriction enzymes with overhangs or sticky ends are more useful because, again, they can form hydrogen bonds, okay, to the nitrogen base that you will add depending on the sequences that are produced in the overhangs or in the sticky ends. Okay? Another one, type 3 restriction enzymes. So for type 3 restriction enzymes, they resemble type 1 enzymes in their ability to both methylate and restrict the DNA. And like the type 1, okay, they are complex enzyme with how many subunits? Usually 2 subunits. Okay? And recognition sites for these enzymes are asymmetrical. Asymmetrical. So what are they going to produce usually? Okay, sticky ends or blunt ends? Okay, sticky ends. And the cleavage of the substrate DNA occurs between how many base pairs? Okay, it's either before, between, or after 24 to 26 base pairs from the side to the 3' end. So, the longer base pairs, okay, can be found in what type of restriction enzymes? Okay, type 1, because they have to scan 1000 base pairs because they before they can cut that specific sequence. Unlike your type 3, they only have to scan 24 to 26 base pairs, okay, before they recognize that specific site. Okay, so example, CG, GC, okay, AT, ATTA. Okay, that's an example of type 3 isoenzyme, Hind F3. Okay, so this just shows you, okay, the differences between the restriction enzymes. I just got this picture from the internet, okay, for easy memorization. So one of the differences of which is the type 2 doesn't use this ATP. Okay, both of them uses magnesium also. Okay, and this is the most frequently used. Okay, so this is the summary of the function of the restriction enzyme. So it will scan first, okay, the length of the DNA. 1000 for type 1, 24 to 26 for type 3, okay? Palindromes in type 2, okay? Or how do you call this? In type 2, usually symmetrical, okay? Because in type 3, it's usually asymmetrical. Okay, and then once they scan, okay, the DNA, it will identify the specific sequences. And once it identifies the specific sequence, okay, the restriction enzyme will bind to the restriction site. Okay, and again, that's to cut the 3' oxygen atom, okay, in the phosphodiester bonds, okay, in the sugar phosphate backbone, breaking the phosphate group. And since I am talking about magnesium, usually we are talking about type 1 and type 2 restriction enzymes here. Okay, and either blunt or stagger ends will be formed. So that statements, okay, sum up to this one. Scanning, okay, until it recognizes this red okay, recognition sequence. Okay, just for the sake of this discussion, the recognition sequence was colored red. And of course, ends are produced, and in this case, this type is a sticky end. Okay, and this one class, okay, these are just terms and definitions. This is isoschizomers. Isoschizomers and neoschizomers. Okay, isoschizomers, they have same recognition sequence, same cleavage. Okay, for example, GGCC. GGCC, this is enzyme 1 or RE, restriction enzyme 1. This is restriction enzyme 2. They are different restriction enzyme, right? But they have the same recognition sequence, GGCC, GGCC. And they also cleave the DNA in the same cleavage site. Like for example, between G and C, between G and C. So that's isoschizomers. That although they are different enzymes, they have the same recognition sequence and they have the same cleavage site. However, when you say neoschizomers class, they have the same recognition sequence, but what differentiates them from isoschizomer is that they cleave or cut the DNA at different site. Like for this one, it has C, 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 G, G, G. Same with this one, 3 C's and 3 G's. But this one, it cuts between 2 C's, right? This one, it cuts between G and C. Thus, this one, the XMA1 produces, okay, sticky ends, okay, while this one produces blunt ends, okay? Next one, this is just showing you the nomenclature of the restriction enzyme. Again, I know that you know this already and you have several examples in the laboratory, but the first letter, again, just to remind you, it's belonging to the genus, species, okay, that is for the second letter and the third letter, okay, of this of the first letter and the second letter of the species, I mean, okay, like in E. coli, okay, strain R, RY13 strain, and from all the RY13 strain bacteria, what is the order of identification of such bacteria? Okay, so we have first. So this is the first, okay, first RE or restriction enzyme, okay, identified in E. coli RY13 strain. Okay, so I hope that you understand everything, class. Okay, next discussion I will be giving you things about plasmids. Okay, so I hope that you learn, you read ahead of time. Okay, you read your book because that's the only way that you can understand the topic. Okay, do not rely too much on the notes of your teacher because most of the time, I personally will only give those things that I have mentioned and are not found in the notes. Okay, I know that you know that already. So read your books, class. It will be very helpful. Okay, thank you very much.